Hello everyone. So for this lecture today we're going to be talking about some evidence of evolution. So this quote from Theodosius Dobzhansky, who is the godfather of evolutionary genetics, says, "Nothing in biology makes sense except in the light of evolution." And that's really true because a lot of the things that are very core to the biology study really rely on this theory of evolution. And so I want to make it clear that just because something is a theory doesn't mean that it isn't well supported. So a lot of times you'll see maybe, I don't know, NCIS or one of those shows, and the lead investigator will say, "Well, I have a theory about this." And really, what he means is that he has a hypothesis, which is just an educated guess. A theory, in terms of when we talk about it in biology, a theory is one of the most well studied and well supported statements that you could make. So, evolution, the theory of evolution, is really well supported. And today we're going to talk about the things that support it. Okay. And when we talk about evolution, evolution can happen on a large or small scale. When we talk about it happening on a large scale, we're talking about something called macroevolution, macro meaning big and evolution. And when we talk about it on a small scale, we're talking about microevolution. So let's just define what we mean by macroevolution. Like I said, macroevolution refers to large-scale changes that occur over an extended time, such as the formation of new species and groups. So you can see over here, we start off as maybe some sort of single-celled organism, and over a very large amount of time, we're talking billions of years, we have seen a single-celled organism, the last universal common ancestor. Now we've got all sorts of organisms that are completely different from each other and totally different for sure from the original common ancestor. Now, when we talk about microevolution, what that means is small scale changes that affect just one or a few genes and happen in populations over shorter timescales. So this is just smaller things, changes in our genetic code, changes in genes that we might see in our lifetime or in a very relatively short amount of time compared to hundreds of millions of years and billions of years. But is there a difference? And the answer really is no. Microevolution and macroevolution really aren't two different processes. It's the same process, which is evolution. It's just occurring on different time scales. Microevolutionary processes, which are the changing of genes, if that occurs over and over again, over thousands or millions of years, it can add up to large scale changes that define new species or groups. So there's really no difference. It's just maybe if we talk about microevolution, it's just better stated that microevolution are small changes that you could see over a small amount of time. So let's talk about the evidence. The first is anatomy. There's evidence in anatomy. Species may share similar physical features because the feature was present in a common ancestor. We call those homologous structures. So I'm going to list out all of them, and then we'll go through them one at a time. The next is molecular biology. So DNA and the genetic code reflect the shared ancestry of life. DNA comparisons can show how related species are. Okay, we've talked about how the genetic code is universal. All organisms use DNA as their genetic code, and we can compare the DNA of organisms, and if they're similar, they're more similar. If they're very different, they're much more different. So we'll talk about that. Biogeography, where we look at the global distribution of species of organisms and the unique features of island species reflect evolution and geological change. So if you find a specific organism all over the globe, well, that tells you one thing, and if you only find it on a very specific island, that tells you another thing. And so we'll get into that. The next is the fossil record. Fossils document the existence of extinct past species that are related to present day species. And so we'll talk about the strengths of the fossil record and we'll talk about some weaknesses of the fossil record. And the last one is some direct observation, where we can directly observe small scale evolution in organisms with short life cycles, like pesticide resistant insects, or how about MRSA, which is methicillin resistant Staphylococcus aureus, The increase of MRSA is one example of direct observation of evolution. So this is what we're going to talk about today. Let's get into anatomy. So anatomy as evidence. When we talk about homologous structures, if two or more species share a unique physical feature, such as a complex bone structure or body plan, they may all have inherited this feature from a common ancestor. Physical features shared due to evolutionary history or a common ancestor are said to be homologous. And so that's a great definition right there, but what does it really mean? Take a look at this. Here is a human arm, a dog's leg or arm, if you want to call it an arm, a bird's wing, or a whale's flipper. And what I'm showing you here is that every bone that is specifically colored with this brown and this white, red, yellow, and maybe dark brown color, all of these bones are directly found in all of these other species. They're a little bit modified for their own benefit, for the way that the organism moves, but all of these bones are still found. And so what it can be said is that these are homologous structures. They've evolved through similar pressures to basically contain the same bones. And what you could extrapolate from this information is that any common ancestor between these would have had these same bones as well. Okay. How about if we look at embryology? These are embryos right here of various species: fish, salamander, turtle, chicken, rabbit, and human. And as you can see, they look very, very similar as they start off their development. But as we go on, we start to look pretty different. But even the second stage here, we've got a lot of similarity between the turtle, chicken, rabbit, and human. The amniotes, this fish, reptile, and salamander look a little bit different, and you could tell that makes sense because fish and salamander are a little bit more distantly related than compared to these turtle, chicken, rabbit, and humans. Um, and what I'm trying to point out here is some homologous structures can only be seen in the embryos, and for one really big part of it is the gill slits that you see in all of these. Even humans have gill slits and a tail during these early developmental times, where as a human the tail basically is no longer used. It becomes your tailbone, and we do not have a tail at birth or at adulthood, whereas other organisms can retain their tail. Okay, let's talk about vestigial structures. What are vestigial structures? Sometimes organisms have structures that are homologous. They're the same type of bones to other organisms, but they have lost their major function. Here are some leg bones in a whale that no longer utilizes back legs, but the bones are still there. These structures, they're often reduced in size and are known as vestigial structures. So the tailbone in humans is also known as a vestigial structure because we don't utilize it anymore, and it's very small, the tailbone, and it's no longer something that we see in full grown adults. So those are vestigial structures, just another piece of evidence that this shared a common ancestor that probably did use some back leg bones. So basically the complete opposite of that I want to throw in here are analogous structures. So this is what we call convergent evolution. And what I'm talking about is not all physical features that look alike are marks of common ancestry. So instead, we call them analogous. They evolved independently in different organisms because the organisms lived in similar environments or experienced similar selective pressures. And what that means is wings, for example, they are all used to perform a similar function, which is flying, but their structure is much different. You can see this moth wing has no bones, whereas this dinosaur wing, it does have a small amount of bone, but a lot of cartilaginous structure here. This has some bone and feathers, and this bat wing has bones all throughout with skin and extra features that the bird wing doesn't. So they're all used to do the same process, but their structuring is much different. So we call them analogous structures. If you were to look at this bat wing compared to a human arm, we would see that we have very similar bone structure here. And so we would call a bat wing and a human arm homologous structures, but multiple different wings that are structurally different. We call them analogous structures, so not similar. So they've all grown up in an environment where flying is beneficial to them, and so they've converged. So they've come together to evolve some structure that can allow them to fly. But the way that it works, the way that it's set up, is different. So that is analogous structures that arise through convergent evolution. And so I just put here, these structures are functionally similar, but anatomically different. Okay, let's talk about molecular biology as evidence. At the most basic level, all organisms share the same genetic material, which is DNA, the same or highly similar genetic codes, the same basic process of gene expression, which is transcription and translation. Remember, transcription, we talked about, is going from DNA to RNA, and translation is going from RNA to a protein. And they also have the same molecular building blocks, and to build proteins, we have amino acids. Okay. These shared features suggest that all living things are descended from a common ancestor, and that this ancestor had DNA as its genetic material, used the genetic code, and expressed its genes by transcription and translation. And that pretty much makes sense, but before scientists ever knew about DNA and how transcription and translation worked, this was a big piece of the puzzle that was missing. And so once we figured this out, it really gave a lot of support for basically having a universal common ancestor. If all organisms have this same genetic info, it makes sense that we've all probably descended from that original thing. So just some interesting things to think about is present day organisms all share these features because they were inherited from an ancestor. So if you look at a very small amount of time, you have the features that you have because of your parents and their parents and the great grandparents. Okay, so if you go back hundreds of thousands of years to the early hominids, you share features with them that they shared with their parents and their parents, and it just keeps going back. And we call it descent with modification. So we're all a little bit different than we were 100,000 years ago, and very different than we were billions of years ago. So we just talked about homologous structures. Let's talk about homologous genes, and that pretty much makes sense if you break down the words homologous, same genes. Biologists will compare the sequences of related genes found in different species to figure out how those species are evolutionarily related to one another. The basic idea behind this is that if two species have the same gene, then they inherited it from the common ancestor. And one example of this is humans, cows, chickens, and chimpanzees all have a gene that encodes for insulin. And this gene would have been present in their last common ancestor. So scientists can take the sequences of the two insulin genes and compare them. And so if we take a look at this chicken's insulin gene and a human's insulin gene, we're about 64% similar. And if we take a human's insulin gene and compare it to a chimpanzee's insulin gene, we're about 98% similar. And so the more similar the gene, the more closely related the two species. And that really makes sense because we know that we shared a very similar, a very recent, in terms of evolutionary time, common ancestor with the chimpanzee, much more recent than we shared it with the chicken. So that's just sort of a numerical way to look at it, but very interesting to say the least. Okay, so the next piece of evidence is biogeography. And biogeography looks at the geographic distribution of organisms on Earth. And that follows the patterns that are best explained by evolution in combination with the movement of tectonic plates over time. So an example is that broad groupings of organisms that had already evolved before the breakup of what we call this supercontinent Pangea, where all the land mass was connected when Earth began. That's about 200 million years ago, where it breaks up into the continents that we know today. If those organisms were spread throughout that landmass, they tend to be distributed worldwide now because they were there and then it broke up and they were still there. In contrast to that, broad groupings of organisms that evolved after the breakup tend to appear uniquely where they evolved, right? If they evolved after landmass broke up, they're not going to be able to cross oceans for the most part, and so they're going to be distinctly where they evolved. Here is a good video of Pangea. Take a look. This is the entirety of Earth's landmass connected. So theoretically, if an organism evolved here and it spread out, it could move to every continent, eventual continent that it could make it to. Okay, and here, about 200 million years ago, we see a breakup into a globe that we can recognize today. So, if you've never heard of Pangea, that's what it is. It's a supercontinent. Eventually, some tectonic movement of the tectonic plates separates the Earth into the continents that we know today. If an organism evolves at some point here after this breakup, it's not going to be able to get to South America or North America, and it'll be distinctly found here. All right. One example of this is Australia's marsupials. Almost every mammal in Australia is a marsupial. A marsupial has a pouch for its young to live in. We know we're very familiar, at least in the media sense, with kangaroos and um, uh, so no, really there aren't many marsupials on any other continent. There are a few, but almost 100% of all the mammals on Australia are marsupials, and presumably that's because Australia is very isolated. It's a continent that's completely surrounded by water and pretty far away from any other landmass, and so these guys evolved on their own there, and you don't find them anywhere else because of that. Okay, let's talk about the fossil record, and I want to just put this picture in. As you can see, layers and layers of rock, and as we get closer to the bottom here, the older the layer of rock is. And so that's just a good image to keep in mind when we talk about this. But of course, fossils are the preserved remains of previously living organisms or traces of the organism. It's usually not the entire organism, sometimes it is. And it dates from the distant past. The fossil record is incomplete because not all organisms can be fossilized, and the ones that can, we haven't found all of them. And so it doesn't give us a complete history of the Earth. That would be great if it did, but it definitely doesn't. But it gives us a really good idea and it's a good look at many things. So the fossils that we have collected offer unique insights into evolution over long time scales. We can quite literally see transitional species, which are just interspersed species from what we have now. This is Equus, or basically a horse, and Mesohippus, the very old horse-related ancestor. And the fossil record of the horses are actually really complete, and so we get to see every transitional species up until what we've got today, and that's really cool. Um, Fossils are often contained in rocks that build up in layers, and we call them strata. So those layers of rocks that we saw, they're called strata. It provides us sort of a timeline where the layers near the top are newer, and layers near the bottom are older. We can also, once we get the fossils, we can do what's called radiometric dating, and that's a process that measures the radioactive decay of certain elements, and it could tell you pretty much exactly how old that rock is or how old that fossil is. So that's what the fossil record does for us, and it's one of the best pieces of evidence that we have for evolution. So finally, I want to talk about direct observation of microevolution. Um, Important modern day examples of evolution include the emergence of drug-resistant bacteria like MRSA and the pesticide-resistant insects. And so we'll talk about the insect example in this one. In the 1950s, there was this worldwide effort to eradicate malaria, which is a mosquito-borne illness, and they wanted to eliminate mosquitoes. So we created this pesticide called DDT, which was sprayed broadly in areas where mosquitoes lived, and At first, it was highly effective at killing mosquitoes, uh, not to mention all the side effects it might have on us, but that's another story for another day. Over time, the DDT becomes less and less effective, and more mosquitoes survive, and that's because the mosquito population evolved in resistance to the pesticide. So here's a little example of that. We've got a few mosquitoes are going to have this resistance. This is random by mutation, right? We talked about how different traits evolve in a population. It's through random mutation. So just a few will have it. You spray DDT and it kills the ones that don't have the resistance, but the ones that do survive. And over a very short amount of time, because these organisms can reproduce very fast, these two get together, they reproduce, and all of their offspring have the resistance genes. So that is a very quick and very basic example of evolution in a very short amount of time, but it's very strong evidence for how this stuff happens. So let's do a summary. Um, Basically, there's multiple types of evidence that supports the theory of evolution. Once again, it wouldn't be a theory if it didn't have massive support. Homologous structures provide evidence for common ancestry, while analogous structures show that similar selective pressures, basically an environment that provides a benefit if you can do something, can produce similar adaptations or beneficial features. Similarities and differences among biological molecules, example, the DNA sequence of genes, and how similar they are, can be used to determine our species relatedness. Biogeographical patterns provide clues about how species are related to each other as well. If they are only found on a specific island, that shows you that they evolved on that island. If they're found everywhere, that means they must have evolved prior to the breakup of Pangea. The fossil record, though incomplete, provides a lot of information about what species existed at certain times of Earth's history. Um, some populations, like those of microbes and some insects, evolve over relatively short periods and can be observed directly. Okay, everyone, that is it for this uh, lecture. Look forward to seeing the exam that I'm going to post on Friday. The exam will be posted on Friday, and you will have the entire weekend to work on it. So it will be due Sunday at midnight. It will be on Canvas, and all submissions will go onto Canvas. So if there's any questions, please feel free to email me or get in touch with me on Canvas. Bye now.